Professor, I have a question.
Yes, please. I'm ready for your question.
Okay, so I was just reviewing and I saw like I remember when you said that reflexes are done through the spinal cord, like those reactions are through the spinal cord and I think they're involuntary. Does that have any relation to do with the amygdala? Because I also know that the amygdala is responsible for like fight or flight and like there's like certain like the way you act with that is like it's like involuntary, like you can't really control it.
Right. So the amygdala is more emotion, right? And the emotion has nothing to do with muscles. It's just neurons and feelings. And so it's different from the reflexes. Like the reflexes are very quick um, activation of the sensory neuron and then just goes to the spinal cord and goes right out to the muscle with never making it to the brain. So that means, let's say you put your finger on a flame, you and the reflex, when it gets too hot, you move it right away out. You don't need that stimulus to go to the brain. Can you hear the banging? Because we are in big construction here. Can you guys hear banging on the hammer?
No.
Oh, good. So so the anxiety and all that is emotional. It's different. It's feelings. It's neuron to neuron interaction. It does not involve muscle. Is that clear to you?
Yes. And can I also ask just one more question? Sorry.
Sure.
Okay, so also I know that there was something when the involuntary with like the cardiac muscles that it uses the parasympathetic like
Parasympathetic and
yeah.
sympathetic. Yes.
Like what is the difference between that and then like skeletal muscle being activated through the spinal like nervous system? Like
Right. To
what
the
is the
central
difference?
nervous system. Yes, we're going
Yeah.
to talk a lot about that in the next set of lectures. So, what it is, the central nervous system is under, when you are aware of things, the autonomic nervous system, which is parasympathetic and sympathetic, they control mostly the viscera and the heart and bowel movement and so on, where there's no voluntary control. So it's different. Actually, you're going to see today that. Wait a minute. So that's the major difference. One is under autonomic control, the parasympathetic and sympathetic. We don't have to voluntarily tell the heart to speed up or to slow down. We don't have to tell the gut, oh, move quicker because I want the food to go down faster or something like that. It's under involuntary control.
Thank you so much.
Is that more or less clear? You'll have a
Yes.
much, much better idea after the next set of lectures. Okay, but those were good questions.
Thank you.
Anything else? No? Okay. So today, we're going to stay a little longer so we can have some review. If you have to leave, this is going to be recorded. So here comes this slide again that you must be so bored looking at it. But just to emphasize, now is the time. The exam is now available for you to download. You will not be able to see the exam, you just see the name 2025 Bio 120 Exam 2. So please download it. It's been available for downloading since this morning, 10 a.m. So please do that. Don't wait. Oh, You mean you can't hear the banging? You cannot hear the banging?
No.
Oh,
No.
that's good. I'm happy. Ah, trying to set this up. There's a lot of construction going up here. They're making new classrooms and so on. So that's good, but it's a pain to be under all this noise. Okay. Yes, you'll be tested on today's material. Just looking here. Oh, there we go. Okay, so let's start. Yes, today's material is on the exam. And I wanted to make you aware that when you go to Brightspace and look at the lectures for session 10 to 16, when you open up Brightspace, you will only see the slides and notes up to lecture 14. And then there's a little sign on the left bottom that says more. You have to click on that to see the slides and notes for lectures 15 and 16. So please be aware of that so you don't miss that material. Okay, so please download the exam. And remember to, when you come in, everything has to be working properly. Remember, we don't have any technical expertise on computers and also on ExamSoft. You won't be able to access the technical support while you're taking the exam. So your computer should act like a typewriter because you don't really have to access the internet. So make sure that it's working properly so you have no problems. Because if some of you already missed exam one, you cannot miss more than one lecture exam. So please make sure that you come prepared. And okay, that's it. If you have problems here, you can contact the tech support that is very, very good, and they will help you work out whatever problems you have. Okay, so let's move on to smooth muscle. So we went through skeletal muscle, smooth muscle, and now, skeletal muscle, cardiac muscle, and smooth muscle. And remember that skeletal muscle and cardiac muscle, both of them were striated. So you had the thick and thin filaments. They were arranged in sarcomeres. So sarcomeres, the Z lines that limit the borders of each sarcomere are the Z line. It's made up of proteins, alpha actin, and so on. And they anchor those thick and thin filaments in place. And also you have the M line, remember, in the middle that anchors the thick filaments. Now here in smooth muscles, you have a totally different architecture. You still have the thick and thin filaments. So you still have myosin and actin, but you don't have troponin and tropomyosin. So those are gone. Those are just present in skeletal and cardiac muscle. Here, the protein that binds calcium is known as calmodulin. We will talk about that in a little bit. Okay, so you see that the shape of the smooth muscle fiber is very different. It's fusiform. It doesn't have those myofibrils, and it still has the myofilaments, but they're not arranged in myofibrils. And it has only one nucleus in the center. And you see that it can be arranged in layers. So if you think about the intestine, the gut, it's a tube and it's surrounded by layers of smooth muscle that allow it to move like through peristalsis so that the food goes down the gut. And also remember the blood vessels. So the blood vessels also are lined by layers of smooth muscle cells. And this, remember, these respond to signals coming from the hypothalamus, which is the thermocenter of the body. So the muscles can contract and relax. These smooth muscles that make up the walls of the blood vessels, they respond to signals coming from the hypothalamus. And as one of your colleagues was mentioning, this would go through the sympathetic and parasympathetic nervous system. I want you to see my hands. Let's like this. Yeah. Okay, so where are the major locations of smooth muscle? Blood vessels, I just spoke about it. Respiratory system, so trachea and so on, all the way down. Digestive system, this is very, very important. It goes all along the gut. The urinary system, remember the bladder, it's very, very important. So smooth muscle is present there. Also reproductive system, uterus and so on, and the eye. The eye is very important because if you think about the eye, how the focus is so particular and so precise, because if your eyes are not aligned properly, then everything will be out of focus. Also, if you just have one eye, you don't have 2D vision. You need the two eyes to have 2D vision. So the muscles have to be highly, highly coordinated. And actually, if you look here at the iris in the eye, you see that there are these layers of smooth muscle. Some of them are arranged in a circular manner. These are the circular muscles, and others are arranged in a radial architecture, and they are known as radial muscles. And they respond to different stimuli. So, for example, when there's a lot of light, then your pupil, which is the opening, so this opening allows the sun, the light, the photons to go in, and then they will activate the photoreceptors that are on the retina. So you need the light to get in through the pupil to activate. It goes in and activates the photoreceptors that are at the retina in the back of the eye. So the pupil, if there is too much light, if it's too bright, you cannot focus properly. So the circular muscles become activated and they decrease the diameter of the pupil, and that's the circular muscles. And if the light is very dim, if there's not enough light, then you want the pupil to increase to allow more light to get in. And now here, it's going to be the radial muscles that are going to be activated to open up the pupil. So this is all very interesting how, we don't have to think about this. And we have to thank the sympathetic and parasympathetic nervous system to be able to go through all these phases. Okay, so here's what a muscle fiber, a smooth muscle fiber looks like. Now you see that there's no striation, but you still have thick and thin filaments. But what it's showing here is these structures here, these round cylinders, correspond to the Z disc, or the Z line, I should say, the Z line of the cardiac and smooth muscle. So they are the ones that anchor the thick and thin filaments. So the thick and thin filaments will go from one dense body to another dense body. And these other proteins or filaments that are here are known as intermediate filaments, and they are providing support for the dense bodies. So you see that this is the arrangement of the proteins that play a role in smooth muscle contraction. And you see that when it's extended, it's long. When it contracts, it shortens, like it's shown here. So the size is 2 to 10 micrometers when they are extended. That's the diameter. And they go to 2 to 200 micrometers long. And they have a central nucleus. Now, just like cardiac muscle, they're under involuntary control. We cannot shut our eyes, close our eyes, and tell the heart to beat, or tell the smooth muscles in the intestine to work, or in the blood vessels. So they're under involuntary control by the autonomic nervous system that we will discuss in the next set of lectures. of lectures, and they orthorhythmic. What does that mean? That means that they don't need all of the muscle fibers don't need to be electrical stimulated, and you will understand why. It's a very amazing kind of architecture. So here you have the smooth muscles arranged in layers, but they're not always like this. And this, you have to think that there are smooth muscle layers all along the, the gut, right? So the contraction and relaxation allows for movement of the food down the gut through this mechanism known as peristalsis. So that it forms waves that you see here, the waves that contract and relax and push the food down the gut. Okay, so now how is the arrangement of the release of neurotransmitters? So here it's very, very different from skeletal muscle. Here's the skeletal muscle where you have the neuromuscular junction. Remember what that is. Neuromuscular junction is where the motor neuron meets the muscle fiber. And you see that you have the axon terminals and the axon, actually the axon terminal here is in very close proximity to a motor end plate, and this is the arrangement that you are already familiar with the skeletal muscle. Now here in the smooth muscle is totally different. Here we have been talking about the sympathetic and parasympathetic. So let's say that this is a motor axon, just like this, the axon of a motor neuron. And instead of having these partitions, it has this bead-like structure that are called varicosities. So each of these little circles contains the synaptic vesicles. So here the synaptic vesicles are located at the motor end plate. But here you see that the motor end plate. Did I say that correctly? Can anyone correct me? Okay, so at the axon terminal, the motor end plate is the area in the muscle fiber that's right under the axon terminal. Okay, so here you see a motor neuron, just like this one. And instead of having these axon terminals, you see that it has these varicosities. And in blue, this represents a sympathetic motor neuron, and here a parasympathetic motor neuron. But they all have these varicosities. And what is the advantage here? So you can see that when the electrical signal comes through, instead the varicosities allow the synaptic vesicles will fuse with each of these varicosities and release the neurotransmitter, just like if it would be a shower over this upper layer, the upper layer of the smooth muscle, the upper fibers, they will receive the neurotransmitters. So it's a very different way of releasing the neurotransmitter. Here you have all these varicosities. When the signal comes through, the electrical signal, the action potential comes through, all these varicosities will release the neurotransmitter like a shower. So they will affect many fibers at the same time. Because it's very important. When you have a layer of smooth muscle, you want all the muscle fibers to contract and relax at the same time. So that's the advantage of having this arrangement with the varicosities. And here you see that the neurotransmitters that are in these varicosities, some will contain acetylcholine, like in the motor neuron or in the skeletal muscle, but others contain other neurotransmitters, such as norepinephrine. You will learn this in the next set of lectures. So here, the neurotransmitter is not always acetylcholine. Okay, now, how these motor neurons meet the muscle fibers? How do they innervate these smooth muscle fibers? So here you see that here's the motor neuron that is coming, and you see the varicosities, and the varicosities can release the neurotransmitter over more than one muscle fiber. But then the muscle fibers that are below this upper layer, they're not going to get a direct innervation by these motor neurons. So how the sodium and potassium will leak through the gap junctions. So here the gap junctions play a very important role, just like in the cardiac muscle, that they also have gap junctions to allow for the propagation of the signal to go from one muscle fiber to the other. And the advantage of this here, as I said, is in this case, you have a single unit innervation. That means you have all the muscle fibers are going to respond to the signal coming from one motor neuron. They contract as a unit, a single unit. Here, like if you think about the eye, then the muscle fibers, smooth muscle fibers in the eye, have to be highly regulated to focus, to provide for a good focus, accurate focus. So each smooth muscle fiber receives its own innervation. You see here the varicosities. So this is a multi-unit because you have each smooth muscle fiber will contract and relax almost independently of the signal. Okay, so here again, this shows, I like this slide a lot because it really gives you an idea of how the synaptic terminals here that come down the motor neuron, and the synaptic terminals are these varicosities. And you see when the action potential is transmitted through these varicosities, then they release the neurotransmitter, and the neurotransmitter is released like a shower over the first layer of muscle fibers. So you see that one group of varicosities, so this would correspond to one motor neuron, can control several muscle fibers. You don't have such a one-to-one relationship. So then these muscle fibers that are deeper, then they are not going to be receiving the neurotransmitter directly. The action potential and that is generated in the upper layer of the smooth muscle is transmitted to the lower smooth muscle fibers through the gap junctions. Remember what the gap junctions are. They're channels made by connexins. Connexins are the proteins that make up the channel, and that open and allow the sodium and potassium ions to go in and out. So the network here in the single unit innervation, the network receives a single innervation. So you have a coordinated contraction. And it's very important for the contraction and relaxation of smooth muscle that lines the gut and the blood vessels also. In the eye, for example, you have a multi-unit innervation, so that each fiber is almost individually innervated. So you have much fewer gap junctions. So that means you see here, each muscle fiber receives the varicosities, is in close proximity to varicosities. Okay, so here you see an upper view of the varicosities that look like these sort of beads. And you see here a high mag with mitochondria, and here are the synaptic vesicles. So this is still a motor neuron, but now it's known as an autonomic motor neuron because it's part of the autonomic nervous system, the one that we can control voluntarily. The one here is a somatic motor neuron, or the motor neuron that will innervate skeletal muscles that are under voluntary control. Here we have an autonomic motor neuron, still a motor neuron because it's really bringing the signal from the central nervous system to the effectors. Remember, the effectors are the muscles here. Smooth muscle in this case, skeletal muscle. These are the effectors. So you have the motor neuron. These motor neurons have the cell bodies in the spinal cord. And you see here the first layer of the smooth muscle that is in close proximity with these varicosities. And now here's the multiple unit innervation. So there's a question here. So the varicosities fit in between the muscle fibers, or do they enter inside them? No, they don't enter inside. There's just juxtaposition, right? There's a little space. The synaptic cleft, remember? Synaptic cleft. So they are in close proximity because then you have the here you have the neurotransmitters being released. Oh, here, you see? So here's the synaptic cleft. So they don't go into the muscle fibers. There's this space, and the varicosities will release the neurotransmitter, and then it diffuses and binds to its receptors on the smooth muscle fibers. Okay, so now what is the arrangement of the thick and thin filament? You still have here the sliding of the thick and thin filament. The principle is the same. You still have actin and you have myosin heads. But now the thick here are represented the thick and thin filaments. So the thick filament is represented here in purple and the thin filament in red. And you see that they are attached to the dense bodies. So here are the dense bodies that are equivalent to the Z disc. Sorry, not Z disc, Z line in the sarcomeres of the cardiac and skeletal muscle. So, but you also see here these other filaments that support the dense bodies, and these are intermediate filaments. So the arrangement is different, but the principle of muscle contraction is still the same. You still have the sliding of thick and thin filaments. They don't change in size. And here you see this in greater detail, the dense bodies and intermediate filaments. The dense bodies are made up of alpha actin, and you see the intermediate filaments that stabilize the dense bodies, and you see the thick and thin filaments. So, what are varicosities? So varicosities are these structures, bead-like structures that store the neurotransmitters. Okay, so you see here the sliding of the thick and thin filament. So these slides are really just to give you different views of how the smooth muscle smooth muscle contraction and relaxation still depends on the sliding of thick and thin filaments. And then you see the gap junctions that allow the fibers that don't receive direct innervation from the varicosities, they will still contract and relax in response to that action potential. Okay, now let's look inside the smooth muscle fiber. How is that excitation converted into contraction, right? Excitation, contraction. So here you have the action potential traveling on the sarcolemma of the smooth muscle. And at the smooth muscle, you have voltage-gated calcium channels, which will open and release calcium into the cytoplasm or sarcoplasm of the smooth muscle fiber. So here, just like in cardiac muscle, if you remember, there are two sources of calcium. In the skeletal muscle, the calcium came from the sarcoplasmic reticulum. But in cardiac muscle and smooth muscle, you have two sources of calcium, one coming in directly from the extracellular fluid. So this is the sarcolemma, the plasma membrane of the smooth muscle fiber. It has these voltage-gated calcium channels. So the action potential is traveling along this, reaches the voltage-gated calcium channels, and calcium will go in. Now, also, you have the calcium from the sarcoplasmic reticulum also has calcium channels that will open in response to the signal. So you have two sources, two ways of getting calcium into the sarcoplasm or cytoplasm of the cell. Okay, so now we have lots of calcium in the cytoplasm. What is the next step? There's no troponin and tropomyosin here. Remember, troponin was the protein that bound calcium, and then that caused a change in structure. Oh, hmm, okay. Oh, people in the waiting room can't see that. Let's see. Oh, oh, and two up high. I don't see. I don't see anyone waiting. So I don't know. I think
They're in now.
oh, now yes. Waiting room. Okay, now yes. Okay, thank you. Okay, so oh, hang on. Synaptic vesicles on that motor end plates then. Yes, you have synaptic vesicles, but they are in the varicosities. What is the difference between sympathetic and parasympathetic neurons? Very, very good question. So they are activated under different circumstances. Parasympathetic is activated when you are calm and relaxed. The sympathetic nervous system is activated when there is emergency, so flight or fight. So let's say when you're nervous and your mouth is dry and you have to go to the bathroom often, that's your sympathetic nervous system being activated. The parasympathetic is when you're calm and there's no emergency situation. So it all depends on which neurotransmitters are being released. And we will go into great detail into this when we study the nervous system. Can you repeat what a gap junction is? So gap junction is a channel-like structure that is, let me see here. What do we have here? Gap junctions. You see that they are channels that made up of proteins known as connexins. And these channels allow sodium and potassium to travel from one muscle fiber to another muscle fiber to conduct the action potential, to allow the action potential to travel through the lower layers of the smooth muscle structure. Does it open people? Does it open sodium potassium? Yes, so the neurotransmitters have the same effect on sodium and potassium channels and calcium channels. Yes. So first you have the sodium channels that that induce depolarization. So remember, going from -90 to plus 30. And then you have repolarization going from plus 30 back to minus 90. And that's the potassium that is potassium channels that are open and allow potassium to leave the cell. Okay, so we were here. So let's dive into the smooth muscle and let's see what induces the sliding of the thick and thin filaments. So we have one factor that we already took care of, which is the increase in calcium. Now we need to activate the myosin heads, right? So since we don't have troponin and tropomyosin, how is this calcium signal going to be transmitted to activate the myosin heads? So here you have a new protein known as calmodulin. Calmodulin, cal for calcium, modulin, modulates. Calmodulin, modulates. So no troponin. So calmodulin here represented will bind calcium. When it binds calcium, it activates this kinase, this protein that is a kinase. Remember, kinase is a protein that phosphorylates another protein. This protein, MLCK, the name is myosin light chain kinase. Myosin because it's going to activate the myosin heads of the thick filament. So you need calmodulin, you need calcium to bind to calmodulin, for calmodulin to be active, and then activate in turn myosin light chain kinase, which is here. This is the inactive MLCK, myosin light chain kinase. And when calmodulin binds to it, it activates it. And now this is going to phosphorylate the myosin heads inactivated. So it's a different mechanism, but the result is the same. It's still dependent on calcium. It still activates the myosin heads. But in between, there's a difference. Instead of troponin and tropomyosin, now you have these two proteins that work in synchrony, the calmodulin and the myosin light chain kinase. Okay, so here you have it again, calcium coming from the extracellular fluid. This represents a smooth muscle cell. You have the calcium channels, you have sarcoplasmic reticulum also with calcium channels. They release calcium. They allow calcium to go into the sarcoplasm or cytoplasm of the smooth muscle cells. Calcium binds to calmodulin. Then when calmodulin is loaded with calcium, then it activates this myosin light chain kinase, which is now going to phosphorylate the myosin head. Okay, so now how are you going to get rid of this? You have, how can you induce relaxation? So this is contraction. Now, how do you induce relaxation? So you need to remove the phosphate group from the myosin head, and this is by this other enzyme, which is known as a MLC phosphatase. So a kinase adds a phosphate group, a phosphatase removes the phosphate group. So you have an MLC phosphatase that removes the phosphate group, and then you have a way of getting rid of calcium so that relaxation can occur. So let's see this. Now, question, why is it that for skeletal muscle, you just have one source of calcium, which is a sarcoplasmic reticulum? But in smooth muscle and cardiac muscle, it comes from the extracellular fluid and the sarcoplasmic reticulum. And why is that? Because the length in time, the time of the twitch is much longer in cardiac muscle and smooth muscle. So what, remember the twitch. So you need more calcium to maintain this long spread of twitch time. So you see here, remember this is a twitch, represents the contraction and relaxation of a muscle. It doesn't matter what type of muscle. There's a twitch for smooth muscle, a twitch for cardiac muscle, a twitch for skeletal muscle. And you see here on the x-axis is time in seconds. On the y-axis is the tension, the force that is developed by the muscle fiber. So you see here in the skeletal muscle, it goes up and down, it contracts and relaxes very, very quickly, very few milliseconds. Now, the cardiac muscle is longer. The twitch is longer. Remember, it's important because now, remember that all the cardiomyocytes are contracting or relaxing in synchrony. And what is important is you have to give the heart enough time to either fill with blood or to expel the blood. So you need this this twitch, the muscle contraction, relaxation cycle to be slower. Now look at the length of the smooth muscle. It is very, very much, much, much longer. So it contracts, it stays contracted for a long time, and then it relaxes. And now this is important also because think of the bladder, right? So the bladder, the muscle contraction there is activated by stretch receptors. So you want to allow the bladder and the stomach in this case also to open up, right? To allow it to be filled either with urine or food. And so you don't want it to, as soon as it has a little bit of urine or a little bit of food, to contract and relax right away. You want to give it some time to allow it to fill. So that's why the twitch in the smooth muscle is so long. So the contraction and relaxation cycle is much, much longer. So there's a question here. So skeletal muscle only has calcium from sarcoplasmic reticulum, correct? And cardiac and smooth muscle get from the extracellular fluid, exactly. Or does cardiac also only get from? No, cardiac and smooth muscle, they both get calcium from the extracellular fluid, in addition to sarcoplasmic reticulum. Okay, I'm checking the waiting room. Thank you. Okay, so smooth and cardiac, they both rely on extracellular fluid, the calcium coming in from the extracellular fluid, and calcium coming in from the sarcoplasmic reticulum into the cytoplasm. Skeletal muscle, only sarcoplasmic reticulum. Okay, so now we go to the relaxation phase, right? So we need to get rid of calcium and we need to deactivate the heads of the myosin. So here you have the sarcolemma, and you see that at the sarcolemma, you have this sodium calcium antiport. Remember that we talked about this when in the first few lectures when we were talking about the plasma membrane and how the ions were transported across the plasma membrane. We talk about these antiports that require that use the kinetic energy of sodium to move in the opposite direction from low to high concentration. So this allows for calcium to move out of the smooth muscle cell. And also here you have a calcium pump and you have another calcium pump. So these two pumps are directly dependent on active transport. So the calcium is removed from the sarcoplasm through pumps, calcium pumps that pump calcium out into the extracellular fluid and calcium into the sarcoplasmic reticulum. And then you have the calcium, sodium calcium antiport. And now here you have the myosin phosphatase, which dephosphorylates. It removes the phosphate group from the myosin and therefore allow for relaxation. So you have the phosphatase and myosin phosphatase and the myosin kinase that work in opposite direction. The kinase is activated by calmodulin having calcium. It activates the kinase. The phosphorylates the myosin, and that's what activates it. In the case of relaxation, then you have a phosphatase, another enzyme, which removes the phosphate group and allows the myosin head to separate from the actin and cause relaxation. Okay, so another very interesting property of the smooth muscle is that it has this property that's known as the stress relaxation response. And I already explained to you. So you want to allow the stomach to stretch, 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 and the bladder to stretch, stretch, stretch before you induce contraction and the release of urine or food into the rest of the gut. So this capacity of the smooth muscle to remain relaxed until a certain level of stress is particular, is known as tone, and is specific for smooth muscle. And what is interesting is that these stretch receptors then, they send signals to the brain. And this is what controls the hunger. So they are in the brain, in the brain stem, here, the medulla, the lowest part of the brain. They are known as satiety center. The satiety center is activated and it will give you the feeling of full. You say, oh, I'm so full. That's your satiety center that receives a signal from the stomach. So the stomach speaks to the brain. So there's a question here. So contraction uses calcium and kinase to form ATP, and relaxation, not to form ATP, to activate the myosin head, right? And relaxations uses the phosphatase to remove the phosphate group, not form ATP. No, it has nothing to do with ATP and ADP. Sorry, it's the myosin head, right? The myosin head that is important here, that is being phosphorylated and dephosphorylated. Okay, so this is a very, very important property of smooth muscle. Okay, so I told you that the smooth muscle has this autonomic, has this, it's orthorhythmicity, but it can be regulated just like the heart, the cardiac muscle. It can also be regulated by nerve impulses. So the sympathetic and parasympathetic nervous system can, for example, regulate the heartbeat. So it can beat faster when it is the sympathetic that is being activated, or it can relax, smooth down when it's the parasympathetic that is being activated. And this all depends on what are the neurotransmitters that are located within those varicosities, and also the receptors present on the muscle fibers. So all this is maybe a little bit complicated at this time, but you will understand better. So the signal between speeding up the heart or slowing down the heart depends on the sympathetic nervous system or the parasympathetic nervous system being activated, and they have their specific neurotransmitters. Okay, so smooth muscle can also be regulated by hormones such as epinephrine. Epinephrine is released from the adrenal glands. Remember the adrenal glands or the small glands, there's a pair, and they're above the kidney. You know, I was thinking the other day, why do we have a pair of kidneys? We have a pair of lungs, we have a pair of adrenal glands, but we only have one heart. I was wondering why. Because if we would have two hearts beating at the same time, that would be very hard to coordinate, I guess, in terms of evolution. But it's a pity that we don't have a spare heart, that when one doesn't work properly, then the other one might be useful. But we only have one. Okay, in stretching, can also, so it's a local factor. Stretching also regulates the smooth muscle contraction and relaxation. Okay, so now let's move on to muscle regeneration. So remember, muscle fibers are highly specialized cells, and most of them lose the capacity of undergoing mitosis because they're so specialized, their proteins, their architecture, their properties, that they lose the ability of undergoing mitosis. So, how can you repair muscles? So, for example, let's think about skeletal muscle. Skeletal muscle, they can be damaged, but near in the very close proximity to the smooth muscle, to skeletal muscle fibers, which is represented, one is represented here. You see here these green structures are the satellite cells. So these cells can undergo mitosis and they can fill in the gaps and they can fuse together to form new muscle fibers and under very seldom circumstances. But if the satellite cells cannot work properly, then the muscle tissue is replaced by fibrosis, and that's not a very good um, thing because then it doesn't have elasticity like the muscle fibers. So the skeletal muscle fiber does not regenerate, but it's helped by the satellite cells. What about smooth muscle? Well, smooth muscle, because it's simpler in a way, um, some of smooth muscle cells still can undergo mitosis. So in the blood vessels, there are these cells that are known as pericytes, and they are stem cells. Let's say they are precursors of smooth muscle, so that they can give rise to new smooth muscle cells at the blood vessels. And in the uterus, during pregnancy and so on, it extends. So the smooth muscle fibers there can also undergo mitosis. So that means that the muscle fibers usually escape the cell cycle, right? That's what is represented here. S phase, or let's say G1, S, G2, and M mitosis, and then they exit the cell cycle and go into G0. That means they lost the capacity of undergoing mitosis. This is what happens for um, skeletal muscle and cardiomyocytes, as you will see. But in the case of smooth muscle, under certain conditions stimulated by different factors, as shown here, they can go from the G0 back into the cell cycle. Okay. Okay, so now let's go on to the cardiac muscle. So cardiac muscle cells do not regenerate at all. And so there's a big effort to have cells from the skin being removed from a patient. And can you now put them on a dish and differentiate them into cardiomyocytes so that they can be replaced, the ones that are damaged in the heart? But we are not there quite yet. So the cardiac muscle fibers, also the neurons, they are permanent cells. They are permanently in G0 phase. They cannot go back and undergo mitosis. So the recording will probably be posted tomorrow morning. I will ask if they can give it to me today still. Okay, so homeostasis, remember body temperature when we studied the feedback mechanism? So that is controlled by the control center is the hypothalamus. So you have smooth muscle, which lines the blood vessels, and these fibers, these smooth muscle fibers can dilate, increase the diameter of the blood vessels or decrease. And this contraction is regulated by signals coming from the hypothalamus. So the hypothalamus sends signals through those motor neurons, autonomic motor neurons, to the smooth muscle fibers that line the blood vessels. And the skeletal muscle also contributes to maintaining body temperature, to homeostasis of body temperature by shivering. So if it's very cold, the hypothalamus can also send signals to the skeletal muscle. And when you shivering because it's so cold, that allows for some heat to be generated because of the ATP that is being hydrolyzed. Okay. Remember, ATP is hydrolyzed during muscle contraction and it releases energy. And that energy, if you're shivering, you have your muscles contracting, then a lot of ATP is being hydrolyzed and provides for heat to maintain body temperature. So, let's see here. Okay. So we'll do the exam review today after class. I'm almost done. So is the ability of smooth muscle cells to divide related to a woman's period? Yeah, in a way, it's regulated there also. So skeletal fibers regenerate easily? No. Skeletals, they don't regenerate. Remember, they have the satellite cells. The skeletal muscle, they don't regenerate, but they have the smooth muscle, the satellite cells that can provide for some kind of, you could call it regeneration in a way, but it's not the muscle fiber per se that regenerates. It depends on the differentiation of these satellite cells into skeletal muscle. Correct. Smooth muscles have a harder time generating. Cardiac don't regenerate. No, no. The smooth muscle cells, they can regenerate, smooth muscle regeneration. So the parasites can differentiate into muscle fibers. And also in the uterus, the smooth muscle fibers have some ability to undergo mitosis. So let's say in terms of can regenerate, cannot regenerate. Smooth muscle, the most, skeletal muscle, intermediate, and cardiomyocytes, none. So it goes from smooth, skeletal, and cardiac. Okay, good question. Yes, the review is recorded. Skeletal and cardiac don't regenerate. Smooth muscle a little bit. No, the satellite cells are with the skeletal muscle, not smooth muscle. The skeletal muscle depends on the satellite cells. Right, correct. So fibrosis is when the skeletal muscle, the satellite cells don't do their job. So instead of having muscle fibers, you have fibers that lose their elasticity. And so the muscle that has fibrosis will not be a functional muscle. Okay, so I'm going to go through, let's see, the last 10 minutes. I'm going to go through some muscle disorders just to give you a hint. So, for example, fibromyalgia, this is a feeling that your whole body you have muscle pain throughout the whole body, especially in the lower back and here in the calves. And the reason is not known. It could be an autoimmune disease. We don't know why. And these these patients suffering from fibromyalgia, it's a very, very uncomfortable situation because they suffer from chronic pain. So they have to take medication in a chronic manner, and that's not very good. So there's a big effort to develop painkillers that can be useful for these patients that suffer from this fibromyalgia. Then you have muscle dystrophies. Dystrophies means degeneration. So that means that it's a group of disease. There's many different types of muscular dystrophies. And what happens is that the muscle degenerates because there's mutations in certain proteins that make up the muscle fibers. And I show here Jerry Lewis. He was a very, very funny comedian. And he always had a fun, once a year, he had a fundraising for this muscle dystrophy disorders, which affects children. Usually they are in a wheelchair by the age of 10. No, ALS is not like a muscle. Well, this is a different, muscle dystrophy is different from ALS. So in ALS, amyotrophic lateral sclerosis, there is muscle degeneration not because of the muscle itself, it's because of the motor neurons that degenerate. So remember that phrase, if you don't use it, you lose it. So if the motor neuron that innervates specific muscle fibers, if the motor neuron is not working properly, then the muscle fiber will degenerate. But here, in ALS, amyotrophic lateral sclerosis, it's related to the degeneration of the motor neuron. Here, the degeneration is the muscle fiber. So let's say, for example, one type of a muscle dystrophy is Duchenne muscular dystrophy. And this occurs mostly in boys. It's a hereditary condition. And the gene which encodes for dystrophin, remember the protein dystrophin, I will tell you what it is, but the gene that encodes for dystrophin is on the X chromosome. So usually, or in most cases, in females, there's one copy of the correct gene and the other one mutated, so they don't suffer this condition. Oh. Okay, we're not in the practice question, so I'm not going to go there yet, okay? Yes, fibrosis is the replacement of damaged muscle fibers with connective tissue and decreases the flexibility. Perfect. We will go through the questions soon. Just give me some time, please, to finish here. So here you have dystrophin, right? So the mutation in dystrophin. What does this protein do? If you remember, dystrophin is a protein that links the fin filaments, the actin, to the sarcolemma. So here's the sarcolemma, the plasma membrane of the muscle fiber, and here's the actin filament. And dystrophin is this protein that stabilizes actin. So if it's not working properly, if it's mutated, and it cannot stabilize this structure, then the muscle will degenerate. And these children at the beginning, they're fine. But then as they grow, you see that the muscles start degenerating and eventually they have to be stuck on a wheelchair. So how do you treat this? Well, the best way is to, if possible, to do gene therapy. But there are other medications that can decrease inflammation and so on, but there's no cure. Okay, myasthenia gravis, that's a very interesting disease that is specifically caused by, it's an autoimmune disease, and it's specifically caused by the destruction of acetylcholine receptors. So what is shown here? This is a motor neuron, and here is the muscle fiber, the motor end plate with the acetylcholine receptors, right? So you have, when the action potential is traveling, you have the acetylcholine released and binds to its receptors. And then you have the action potential being transmitted and converted into contraction. But it's an autoimmune disease. So that means that our white blood cells will destroy the acetylcholine receptors on the motor end plate. So that means that these muscles cannot react. They cannot be activated. by acetylcholine because there's no receptors, or there's much fewer receptors. So here you see, again, this is common in females. Autoimmune disease are common in females, and affects neck and face muscles. Here you see the lid of the eye that is not functioning because the acetylcholine receptors are being destroyed. And they're not destroyed in the whole body, that would be disaster, but in certain areas. So how do you treat this? One way is to use inhibitors of acetylcholinesterase. Remember, this enzyme is the one that metabolizes acetylcholine. So by preventing this enzyme from destroying acetylcholine, you'll have more acetylcholine to bind to the few receptors that are spared. Or you can try to calm down the immune system because this is caused by activation of the white blood cells. We don't know why they are being activated against our own proteins, but you want to calm down the immune system with corticosteroids and removal of the thymus. The thymus is a gland that produces antibodies. Okay, so now some toxins. So trichinosis, this is caused by a parasite. This parasite is known as Trichinella spiralis. So this name spiralis because the parasite has the shape of a spiral. So this parasite can be transmitted from meat that is not well cooked. So it can go right from the pig into humans, and it enlarges itself in the paramecium and endomecium. And this can cause very discomfort, diarrhea, weakness, and muscle pain. So it's caused by this parasite. Then you have two diseases that I want to talk about caused by bacteria infection. So this one is known as botulism, and it's a toxin that is produced by this bacterium known as Clostridium botulinum. So this bacterium that is shown here produces a very, very, very toxic chemical, one of the most toxic chemicals that's available. And what does it do? It prevents the synaptic vesicles from fusing with the plasma membrane. So that means that there's no neurotransmitter, no acetylcholine is being released. So that induces paralysis, right? So if you don't have acetylcholine being released from the synaptic vesicles, there is no way that you can activate the skeletal muscle. So this causes paralysis. In this case is known as botulism. But now, believe it or not, this is used to prevent wrinkles. Okay, so this is known as Botox. So what is Botox? Botox is the chemical, the toxin produced by this bacteria. So you have to use it in very, very minute amounts. And wherever you inject it, it's going to paralyze those muscles. And wrinkles are made by the contraction of the overcontraction of muscles in that specific area where the wrinkle is formed. And when you age, then those crevices that are made by the wrinkles don't go back to their original position, so you'll have wrinkles. But in young people, you can have some kind of wrinkles that can be protected by the injection of very small amounts of Botox. Okay, let's see. There's a few questions here. Oh, that's much better. If it produced what's used in Botox, right? Which stops your muscles from contracting. Exactly. You're absolutely correct. Do we have to know all these muscle disorders for the exam? Yes. This is what is in honey for infants. I never heard of that, of botulinum being in in honey for infants. I never heard that. Sorry. So Botox wouldn't have as much impact as an older person as it would on a younger person since it's preventive. Yes, you're correct. Okay. Tetanus. So this is also caused by a bacterium known as Clostridium tetani. And the toxin is known as tetanospasmin. So spasm, it causes spasm, tetanus spasmin. So what happens, this chemical produced by these bacteria inhibits these neurons that are inhibitory neurons. So it prevents these neurons from releasing their neurotransmitter. So the neurotransmitters here are GABA and glycine, and these are neurotransmitters that instead of causing excitation, instead of causing depolarization, they inhibit muscle contraction. And this is very important because think about a car. If you only have the accelerator, you could never stop. And you cannot control the velocity of the movement of the car. So you need a brake. So the same in the nervous system. You have some neurons that are excitatory, that are the accelerators, but you have other neurons that are the brakes, the inhibitory. So you need both. And in Parkinson's disease, for example, the inhibitory neurons degenerate, and that's why you have sometimes tremors. So this toxin produced by the Clostridium tetani induces convulsions because there's no mechanism to prevent muscle contraction. The motor neurons, the inhibitory neurons, are not working properly. Okay, now we have polio. Polio is caused by a virus, the polio virus. And this was devastating. In 1916, there was an epidemic in New York City, and there were 27,000 polio cases. And it could affect even the respiration, that the patients had to be connected to an artificial respiratory machine. And there were 9,000 deaths, nothing compared with COVID, right? So Jonas Salk was the one that discovered the vaccine against polio. And that vaccine was incredible because what happened is the virus was sort of cooked, boiled, so all the proteins were destroyed, and then you injected that into the individual, and the individual would generate antibodies against the virus, and the virus proteins would not have an effect anymore. So, so the idea is you inject the viral proteins that are destroyed. The body makes antibodies against those viral proteins. So when you are infected by a live virus, the body already has these antibodies that will bind to the live virus and will prevent it from activating and from dividing. So Salk was a CUNY student. He was the son of Russian immigrants, and he wanted to go into law school, but then he decided to go to medicine. And thank God, because he discovered the vaccine for polio. You have here some diseases, some conditions, let's say, that are associated with abnormal contractions of skeletal muscle, spasms, tremors, and so on. These two disorders are affected by the lack of control of muscle, of central control. The CNS, the central nervous system, is damaged either because there's not enough oxygen during childbirth or after birth. And if the pregnant woman is infected by this virus, this can lead to cerebral palsy. Palsy means paralysis. And here, the area that is affected is an area that is present in the brain and that affects the opening and closing of the eyes. So here is a problem of the central nervous system. Some of the neurons are not working properly. Now, muscle tumors are very, very rare. And if they occur, they're very aggressive. They metastasize very quickly. Then hernias. So hernias are developed by over using weights that are too heavy. So therefore, you make a lot of force against openings that might be in the body. For example, an inguinal or hernia. What happens here? You have a canal, an opening between the abdominal on the abdominal wall, where the testes will come out and go into the scrotum. So that opening stays there. That's the inguinal, the opening to the inguinal canal. So if there's too much pressure exerted on the walls of the abdomen, sometimes the small intestine can protrude through the inguinal canal. And this, can you imagine, the muscles in the abdominal wall can exert a lot of pressure on the intestine and cause huge amount of pain. So you have it here. So this would be an inguinal hernia. Then you have a hiatal hernia. What happens here is you have an opening also in the abdominal wall where the esophagus goes through and connects with the stomach. So if there's too much pressure exerted in the abdominal wall, also part of the stomach can protrude out of the abdominal cavity, and this is known as a hiatal hernia. Sometimes you have to be operated to release the pressure. Okay, then these are damage to the ligaments or sprains, damage to tendons or strains. And what happens? How can you treat this? Or you use this kind of condition called rise. So you rest, so you don't put pressure on where it is damaged. Ice. The use of ice is very interesting because cold temperature slows down the action potential, the transmission of the action potential down the neurons or up the neurons. So by using ice, you prevent the signal from reaching the pain center in the brain. You're slowing down the electrical current conducted by the pain neurons that conduct the electrical current to the pain center. It also, these three, ice, compression, and elevation, they all prevent swelling. Okay, so the objective of warm up, why do you need to warm up? Why doing excessive exercise? Because you want to, one of the main causes is to have some of the muscle fibers in the muscle to be recruited. So when now you're developing extraneous exercise, you already have some of the muscle fibers warmed up already in the ready to be more efficient. Okay, now this is intramuscular injections. Why do we give intramuscular injections? Because giving chemicals, injecting chemicals into the muscle, they create sort of a bolus there that will diffuse much slower than if you give the chemical right into the blood vessels, right? You give them into the muscle, it forms a bolus, a concentration there, and it diffuses very slowly into the blood vessels. So it's a way of delivering vaccines and other types of drugs that need to have an effect, a long-term effect. Okay, so here are examples of how can we address pain. So analgesics, they're pain relievers. Some of them act in the central nervous system. So there's a center in the brain that where the muscle fibers, the pain muscle fibers, conduct the action potential to that center, and that's when we feel pain, when that center is activated. So if you use some chemicals that can act at the central nervous system or sometimes at the site of injuries, like lidocaine. Lidocaine is a local anesthetic, analgesic. Other chemicals cause muscle relaxation. So like Valium and Datrium. So this is easy to study and to remember. Now here, I know you went through this in the the lab and all this also the levers. I just want to make one comment. Why do we need to study levers and what is the importance of levers? So levers are these machines that can convert a linear force into a rotating force. Now remember, this is very important. So the bone is the lever and it swivels around the fulcrum, which is the joint. So this allows for linear force for the muscle to go around to have a rotating force instead of a linear force. So this design of having the joints that that connect with two bones is very important for allowing for the movement of the muscle not to be just up and down, but can be rotate. So you have the three different layers. Okay, that's it. Let's see what is here. Oh, so relating to the honey in infants, yes, they don't have a gut microbiome to defend against it until they start eating solids. Oh, that is very, very interesting. That's true. I never thought about that. Interesting. Thank you. So Botox wouldn't have as much impact as an older person. Correct. But older people still use it sometimes. Okay. I don't know what I have to do now. Anyway, let's go to some questions. Unless you have questions. Anyone has some questions that you want me to address? Or should I go right to these questions? Do we still have some participants here? I hope so. So, shall I answer these questions? Yes? Someone, please? Yes. Okay. So, let's start with the first one. Okay. Myoglobin. Remember what myoglobin is. Myoglobin is the protein that's only available in muscles, no other cells, and it allows oxygen to bind to it so that it provides additional oxygen besides hemoglobin. So the question is, myoglobin has ATPase activity? No. Is found in thick and thin filaments? No. Accounts for some of the excitability of muscle tissue? No. Binds oxygen within red myofibrils? Yes. That is its function, is to bind oxygen. Rigor mortis. Remember, rigor mortis happens during a 24-hour period after a person dies. And why? Because the body is not making, the cells are not making any more ATP. So the myosin and actin, the myosin is bound to actin, the cross bridges are stabilized, and the muscles stay in contraction. They cannot relax. So rigor mortis occurs after death because of the fusion of sarcolemmas. No, calcium is no longer available. Now, ATP is no longer available for the release of myosin actin cross bridges. This is the correct answer. Okay, the function of calcium ions in skeletal muscle contraction. So function of calcium in skeletal muscle contraction. So what does it do? So it binds to troponin. Troponin changes in structure, sends a signal to tropomyosin. Tropomyosin also changes structure, and it uncovers the myosin binding sites on actin. So that's what calcium does. It makes the myosin binding sites on actin available to myosin. So bind to receptors, no. Cause a pH change, no. Bind to myosin binding sites on actin, no. calcium doesn't bind to myosin, it binds to troponin, right? On the thin filament, so that myosin binding sites on actin can be exposed. So D is the correct one. Tell me if I'm going too fast. If the overall length of a muscle increases during contraction, the contraction is referred to. So the length increases, but the tension is the same. When you're running, or when I'm going up and down, right? So the the size changes, but the tension doesn't. So that means it's an isotonic contraction, right? It cannot be isometric. Iso means the same. It cannot be isometric because it's changing in size. But now it increases in length. It's not decreasing. So if it's increasing, that means it's an eccentric isotonic contraction, right? So first of all, you see right away that it's changing in size, so it cannot be isometric. So it has to be isotonic. Now, if it increases in length, that means it's eccentric. If it would decrease in length, it would be concentric. Okay, so there's some questions here. What was 1 and 2 again? Well, these questions are posted so you can look. Is it on Brightspace? Yes, absolutely. Does anyone know if she, that means me, went over all of the lecture 16, if it's all be in the exam? Yes, all on the exam. What is isokinetic? This is, it's a term that means the kinetic, the movement, the type of movement is the same. Okay, the sustained partial contraction of a portion of skeletal muscle is called trepa, incomplete tetany, simple twitch, tonus, or spasm. Sustained. So that means, what does it mean, sustained partial contraction? So if it would be completely sustained, that would be tetanus. If it is partial contraction, that's incomplete tetanus. Remember, when the frequency of stimulation is so fast that the muscle will not relax, so it will stay at the twitch will be stable, will reach the maximum. So, but it's partial contraction, sustained partial contraction, that would be incomplete tetanus. So the correct here is eccentric isotonic contraction. Isotonic because the tension is stable, just the size changes, and because it's longer, that means it's eccentric. If it decrease, it would be concentric. Which of the following, the answers are at the end. Which of the following is found in skeletal muscle but not in smooth muscle? Well, we just discussed that. So here, which of the following is found in skeletal muscle but not in smooth muscle? Actin, myosin, troponin, right? So troponin is found in skeletal muscle and not in smooth muscle. Instead, in smooth muscle, you have calnodulin. Number 7, the sarcolemma, this is very easy, is a specialized form of plasma membrane. It's the plasma membrane on a muscle fiber. Will we get some questions from this list on the actual exam? You kidding me? No, I'm so sorry. Actually, I think there's one, maybe. I don't remember. If there is one, there's only one. According to the notes, incomplete tetany permits partial relaxation. So I'm a little confused. And it says muscle tone is sustained partial contraction. So sustained partial contraction. Well, partial relaxation or partial contraction, to me, they're almost synonymous, right? Muscle tone is sustained partial contraction. Well, I'm confused here with you, but I think the best possible answer here when you have something. So there's nothing here that says tone. Tonus. Oh, okay. It says muscle tone is sustained partial contraction. Oh, okay. So Samia, you are absolutely correct. Thank you. This is true. So the tetany and trepa, and they relate to relaxation. The tonus relates to contraction. So your colleague is absolutely correct. So sustained partial contraction is the tonus. Thank you. You're going to do well in the exam. Very good. Thank you. So I don't know what I put. Let's see. 5. What's in 5? B. Well, this is incorrect. This should be D. I will correct it later. Thank you, Samia. Where are these questions? These questions are posted on Brightspace near the notes. Yes, the answer is tonus. Yes, absolutely. Okay. Which of the following is found? Oh, we did this, right? We did this one. Now, creatine phosphate. Remember, this is the part of the phosphagen system that is available in muscles and neurons. Is a myofiber waste product? No. High energy compound that can generate ATP? Yes. B is correct. So remember, in muscle fibers, you have the phosphagen system and not anaerobic system or mechanism and the aerobic mechanism to produce ATP. And in the phosphagen system, you have creatine phosphate that donates a phosphate group through the creatine kinase, donates one phosphate group to ADP to make ATP. Samuel, Eugenius. Yes, very good, John. Okay, here we go. Which of the following is the smallest unit? Ooh, myofilament. That's the thick and thin filament. Myofibril, that's those cylinders that are inside the cell. Myofiber, that's the muscle fiber. Fascicle, a group of muscle fibers, and motor unit, that's the motor neuron plus the muscle fibers. So the smallest would be A, myofilament. 10. The function of calsequester. So where is calsequestrin? Calsequestrin is located in the sarcoplasmic reticulum, which is a specialized form of endoplasmic reticulum. So calsequestrin is inside the sarcoplasmic reticulum. Its function is to bind calcium. Remember that the sarcoplasmic reticulum has 10,000 times more calcium than in the cytoplasm. So inside the sarcoplasmic reticulum, you have this protein called sequester that binds calcium ions within the sarcoplasmic reticulum. So A is the correct. Okay. During an isometric contraction. Now let's see, Samia, help us out. During an isometric contraction, isometric, that means it doesn't change size, a muscle shortens and exhibits greater and greater tension. No. Isometric means it doesn't change in size. Lengthens, no. Relaxes and moves by gravity, no. Shortens by maintaining nearly constant, no. E, remains the same length but exhibits increased tension. E is the correct. So iso means the same, metric means size. Isometric doesn't change size, so it remains the same length but increases tension. Good, Samia, thank you. Okay, the function of intercalated discs. Remember what are intercalated discs? Intercalated discs are the areas where two cardiac muscle fibers meet. And these are very special in cardiomyocytes. Why? They have two special structures. They have desmosomes. Remember what desmosomes are? They are the Velcro that keeps the cardiomyocytes very much attached to each other. So when they contract and relax, they don't come apart. If not, the heart would just disintegrate. So they have desmosomes and they have gap junctions so that the sodium and calcium can go through. So the function of intercalated discs is to separate the sarcomeres, no. Generate the appropriate neurotransmitter, no. Store calcium, no. Provide a mechanism by which all the cells in the network can contract as a functional unit, yes, and store ATP. So D is the correct. Isotonic changes length. Concentric isotonic shortens and eccentric lengthens. Very good. Okay. Now, 13. A motor unit is defined, motor unit has the motor neuron and the fibers and the muscle fibers that it regulates, that it innervates. It's a single neuromuscular junction. No, because the motor neuron can be divided into different axon terminals. Single motor axon and all the myofibers it innervates. B is the correct answer. A single myofiber, a single muscle, but all the other ones are incorrect. So motor unit contains one motor neuron and all the myofibers, all the muscle cells that it regulates. You don't get 5. So Samia, ask Samia, she will explain to you. So here, partial contraction refers to tonus. Partial relaxation is the theta. Okay, we went through this already. We passed this. If I have time, I will come back to it. Okay, myasthenia gravis, we just talked about it. It's treated with drugs that block acetylcholine esterase. The purpose of this treatment is to allow acetylcholine to have more acetylcholine at the synaptic cleft so that it can bind to the few receptors that are not destroyed by the white blood cells. So increase acetylcholine concentration. That's A is the correct one. Right away, acetylcholine concentration to offset the partial blocked ACH receptors because they're blocked with the antibodies. Okay. We'll come back to question 5. Let's see how many more we have. 2. When calmodulin, so remember calmodulin is the protein in smooth muscle fibers that binds calcium. It's the cousin of troponin. So troponins cousin is calmodulin. Binds calcium ions in smooth muscle tissue. It causes troponin to move away from the myosin. Smooth muscle tissue causes troponin. No, this is false because there's no troponin. It binds calcium ions in smooth muscle tissue. This part is correct, but it doesn't cause troponin to move away. So the second part is incorrect. So it's false. Overall, it's false. Which is not a risk factor for osteoporosis. So remember, osteoporosis means that there is the osteoclasts are overactive and the osteoblasts are underactive. So what is not a risk factor? Menopause is a risk factor because there's less estrogen. Moderate exercise, that's not a risk factor. Diets deficient in calcium, yes, that's a risk factor. And decrease in estrogen, that's a risk factor. And smoking is a risk factor. So the correct answer is moderate exercise is not a risk factor. So you have to be careful to read the question very, very carefully and slowly as you can. Secondary ossification centers. So we're talking about endochondral ossification, where you have a mold and the ossification goes from the periphery to the inside. So secondary ossification centers, they form in the epiphysis. The primary ossification centers, they form in the diaphysis. The secondary ones form one in each of the epiphyses of the long bones. Diaphysis, no. In epiphysis, yes. Along marrow cavity borders, no. At the edges of primary ossification centers, no. Along perichondrium in the surfaces, no. Okay, during normal homeostasis, blood calcium rapidly decreases in response to, okay, so let's say you have normal homeostasis, normal levels of calcium, and now you have calcium to decrease. Parathyroid hormone increases calcium levels. Why? Because, for example, it activates osteoclasts, so there's more calcium being released. It induces more calcium to be absorbed from the intestine, and it increases the levels of calcitriol. So parathyroid hormone increases calcium levels, does not decrease. The active metabolite made from vitamin D, no. This also increases calcium because the active metabolite is calcitriol, and it allows for more calcium to be absorbed from the gut. Calcitonin, well, that's an option because calcitonin is released when there's too much calcium. So calcitonin would decrease the levels of calcium. Thyroid hormone does not have, and vitamin C don't have that much effect. So the correct answer here would be C. Okay, last slide. All of the following are true for bone remodeling. So remember, bone remodeling, you have osteoclasts that destroy bone, and you have osteoblasts that build bone. They work together. So all of the following are true for bone remodeling, except it continues throughout life. Yes, that's true. It allows the release of calcium to the blood. Yes, because osteoclasts are working. It allows growing bone to respond to mechanical forces. Yes. It results in the formation of new osteons. Yes. It is found only in fetuses. No, it goes through life, through life. So the one that is wrong is E, correct? Okay, osteoarthritis. So osteoarthritis is not, does not have inflammation. Inflammation is not involved in osteoarthritis. It's only the wear and tear. Appears to be related to meat in the diet. No. Vitamin C deficiency. No. Increased physical activity. Yes, it could be because you have too much the bones rubbing against each other and you have the wear and tear of the articular cartilage. Systemic metabolic disease, no. Degeneration of articular cartilage. Yes. Well, here there's two possible answers, right? Osteoarthritis definitely is related to degeneration of articular cartilage. So if there is too much physical activity, like those individuals that overrun and overuse weights and so on, eventually they can suffer from osteoarthritis. So this question has two possible answers, I think. What do you think? Huh, okay. Which of the following is not true of red bone marrow? Consists of adipose tissue? No, that's the yellow bone marrow. Is found in spongy bones? Yes. Produces blood cells? Yes. So the one that's not true is that it's made up of adipocytes. Finally, the procedure for examining and repairing the interior of a joint with minimal incisions is known as arthroscopy. Okay, I think we reached the end. So, Heather, are you still here? Do you want me to go back to question 5? Yes. Okay, so let's go back to question 5. Yes, you can stay on, or the person that wants to speak with me, please let's turn off the Zoom and then you sign on again. Okay, let's go to question 5. The sustained partial contraction of a portion of skeletal muscle. So sustained partial contraction. So that means that the muscle is contracting, right? So it's different from sustained relaxation. It's called tonus, right? So that means every muscle fiber is in some form, light form of partial contraction. Remember? If not, it would be flaccid. If there's no muscle, if there's no tone, then the body would be flaccid. Remember the picture of the little baby that is totally, has no strength, is flaccid. So the sustained partial contraction that is exerted in all muscle fibers, all skeletal muscle fibers, that is known as tonus and not incomplete tetany. Because incomplete tetany should be sustained partial relaxation. So that means that the muscle would relax a little bit. Yes, Heather, you are correct. What is this? Heather, you are correct. But frequent stimulation prevents relaxation, right? So we are talking about the relaxation. So if you have complete tetanus, that means there's no relaxation. But if you have a little bit of relaxation, that would be incomplete tetanus. But here we're talking about contraction. So this contraction is not related to frequency of stimulation. Okay. And actually, also the cue here is that it's a muscle, right? It's not one muscle fiber, it's the whole muscle that is in partial contraction because some of the fibers are contracted. Little relax time, right? So here for 20, let's go to 20. You can be two answers, which would be best for the exam if there's a scenario like that. Well, if there's a scenario like this, I will have to give credit to both answers. But hopefully, I usually go over the exam very, very carefully. But if there's a question like this and there's two possible answers, then I'll give credit to both answers. When will the recording be available? I'm going to ask the person to give it to me as soon as possible, but probably tomorrow morning. Okay, that's it. We're done today. 6 o'clock. Let's go home. I have a cup of tea. Okay, good luck for everybody. Please study. Bye bye. Oh.